Good afternoon all. So here we are with our PGT Microbial World session number 2. So in our previous class we have dealt with the introduction of all the unicellular organisms, all the microbes that we have to dealt with in this chapter, of which we have clearly looked into the structure of tobacco mosaic virus and bacteriophages. So now we will continue the concept with multiplication of bacteriophages. Okay, how bacteriophages are multiplied within the host cell. It is very clear that bacteriophage or virus will not replicate outside the host cell. Replication or increase in their number will take place only within the host cell by utilizing host cellular machinery. So here if you see multiplication of bacteriophages, for virus multiplication it must invade a host cell for sure and use its metabolic machinery. How a bacteriophage will invade the host machinery? If we have to consider this as a bacteriophage, okay, which is infecting the bacteria. At the end of tail fibers, as we have discussed, it has what enzyme? Lysozyme. Okay. So this enzyme will degrade the peptidoglycan layer which is present in the cell wall, and this genetic material is easily dropped into the bacterial genome. Okay. After entering into the host cell, this DNA, that is the genetic material, will start replicating utilizing the host cellular machinery of the bacteria. So this multiplication is of two types. One is lytic cycle and the other is lysogenic cycle. Okay. In lytic cycle, without the association of the bacterial genome, viral genome will use the cellular machinery, it will divide, it will lyse the host cell and it will come out. All the viral particles will come out lysing the bacteria. But then in lysogenic cycle what happens is the genetic material of the virus will be integrated with the bacterial genome. Okay, the genetic material of the virus will be integrated with bacterial genome. This integration of the viral genome and bacterial genome is called as prophase, and it will replicate along with the bacterial genome. That is lysogenic cycle. In lysogenic cycle, bacterial cell will not be lysed, it will not be killed. Okay. So here if we see two multiplication cycles, one is lytic cycle which ends with lysis or the death of the host cell, whereas lysogenic cycle, virus remains inside the host without killing the host cell. Okay, it will remain inside the host without killing the host cell. First if you see attachment or adsorption, phases use tail fibers to attach the complementary receptor sites on the bacterial cell wall. So this will use some complementary receptor sites on where? On the bacterial cell wall, this tail fibers. Second is penetration. The tail sheet contracts, injects the DNA into the host. Okay. Capsid remains outside. Capsid, that is a proteinaceous coat that will remain outside, which is referred as a ghost. Okay. So the host means in which the genetic material is entering. Once it is dwelled into the host cell, this is remained as what? Ghost. Okay. Virus behaves like hypodermic syringe. Okay. Like it is like a syringe attaching over here, injecting this DNA into the bacterial cell. So this is about the introduction of the lysogenic cycle. First is what? Attachment or adsorption. Second is what? Penetration. Third step is biosynthesis. Viral DNA reaches the cytoplasm. Host machinery uses to synthesize what? Enzymes, DNA and capsid proteins for viruses. Okay. Utilizing the host cellular machinery, all the cellular constituents, what are synthesized? Enzymes, DNA and capsid proteins for viruses are synthesized. This initial viral parts are not complete phases, they are detectable. Okay. So this initial viral parts whichever are formed, either capsid is formed, DNA is formed, but all of them are not together at one place. Okay, that individual particles inside the host cell is called as what? Eclipse period. Okay, where not complete phases are detectable, parts, individual components are detected inside the cytoplasm of the host cell. That period is called as what? Eclipse period. Okay. And then maturation. Viral DNA and capsid proteins, both the viral DNA and capsid proteins are assembled into what? Complete virions. Okay. Viral DNA and capsid proteins are assembled into complete virions. Then last step is what? Release, lysis phase. Lysosome enzyme will break the host cell wall, host cell burst lysed into new phase particles, those are virions. This is the stepwise process. First is adsorption, second is penetration, third is biosynthesis, fourth will be maturation, where the individual particles will be grouped forming a complete virion, and fifth is release. This is the lysis phase. Look at the diagram once in the lytic phase. So, can you see the first is what? Adsorption. The pins got adsorbed to the host cell, bacterial cell receptors. Excuse me. Yeah. Okay, they get adsorbed first to the wall layer, and then second is what? Penetration. The DNA genetic material is penetrated into the cytoplasm of the host cell, phase DNA and the host DNA. And then biosynthesis. The phase DNA replicates and the phase proteins are made here. Can you see the individual components like DNA is separate, proteinaceous coat is separate? This will undergo what? Maturation. They will form into a complete virion, and this phase is what? Eclipse period. Eclipse period. Okay. And last, the cells will get lysed and they burst open, releasing out the bacteriophages. This is called as what? Lysis, which is the last stage. So, this is the complete lytic cycle. Clear till here, right ma? Next, lysogenic cycle. So, in lysogenic cycle, can you see here? First is adsorption as usual, wherein fast DNA and the tail fibers will be absorbed at the surface of the bacterial cell. Then the genetic material is injected into the bacteria. This is what? Penetration. And next is this genetic material of the virus will be integrated with the bacterial genome. This is what? Integration. The fast DNA integrates into the host chromosome, forming what? Prophase. So, prophase is formed only in lysogenic cycle, not in the lytic cycle. Of course, we will look into the differences of lytic and lysogenic also, but remember this, that prophase is formed only in the lysogenic cycle. And then after prophase formation, this will start replicating. Okay. Sometimes during some conditions the prophase will not be formed. In that case, from here it will start with cycle, lytic cycle, but when prophase is started, this will continue with what? Lysogenic cycle only. So, the prophase replicates as host divides, it is passed on to the two daughter cells. So, here one cell divided into two, each cell, each bacterial cell is having one prophase. So, can you see any lysis of the bacterial cell here? No, no lysis of the bacterial cell, because each bacterial cell is having one prophase. Bacterial cells are replicating and each bacterial cell is having the viral DNA associated with its genome DNA. Next is induction. When daughter cell is induced, the viral DNA is excised. Daughter cell, when it is inducing, the viral DNA in the daughter cell will be excised, and in the daughter cell synthesis happens, individual particles will be formed, they will assembly or maturation phase, and then complete virion will be formed, and then there will be release of the bacteriophages. This is the lysogenic cycle. What is the only difference that we can see between lytic and lysogenic? In lysogenic, the viral DNA is integrated into the genomic DNA. This integration will result in the formation of prophase. When bacterial cells are dividing into two, in one of the daughter cells, okay, what happens? This fast DNA again is excised out of the genomic DNA. This starts synthesis of the DNA capsid particles. They will get matured, assembled into the complete virion, and they will be released after the complete formation of the mature virions. This is the lysogenic cycle. So, here in this lysogenic cycle, this involves bacteriophages like lambda. Okay, unlike the lytic cycle, no lysis or death of the host cell will occur initially. Okay. In lysogenic cycle, once the daughter cells are formed, from the daughter cell this viral GNA is excised and this will undergo death after the complete virions are formed. That is why this is called as lysogenic cycle. So, here again the same thing, integration, fast DNA is integrated into the host, this is called as what? Prophase, and in prophase this remains latent or inactive, okay, latent or inactive during the prophase. And temperate phases, phases capable of lysogenic cycle are termed as what? Temperate phases. Okay, those bacteriophages which are entering the lysogenic cycle are temperate phases. And replication, when bacterial DNA replicates, prophase also replicates. When bacterial DNA replicates, prophase will also replicate. This passed into the daughter cells during the binary fission. Reactivation, during stressful conditions, for example in UV light or chemicals, prophase may get detached. Okay. During stressful conditions, that is for example in UV light or chemicals, what happens? The prophase will detach and this detachment will lead into the entry of what? Lytic cycle. The primary difference is from formation of prophase in the lysogenic cycle. Second difference is temperate phases are formed and from the daughter cells the viral DNA is excised. Okay? Viral DNA will be excised and then that starts the lytic cycle. Clear, right? And then, so here you can see the comparative cycle between lytic and lysogenic. Just a second. Yep. So here first you see adsorption and then penetration or DNA injection into the host cell. Fast DNA will be replicating. If prophase is formed over here, this undergoes cell division from here. Insertion of prophase integrating into the bacterial genome, this enters where? Into the lysogenic cycle. From the daughter cell, once the fast DNA is excised, it is entering to the lytic cycle again. Okay. This is the cycle of lysogenic, and from here the complete viral genome will get replicated, synthesis of new phases, phase assembly takes place and then host cell lysis, lastly host recognition. Clear, right? So, this is the lytic cycle and lysogenic cycle. The only difference is the viral DNA getting integrated with genomic DNA and then formation of daughter cells. In the daughter cells, the viral DNA is excised and then it enters into the lytic cycle again. Okay, this is the comparative analysis of lytic cycle and lysogenic cycle, very important for us. So, here in this table you observe the comparative analysis between lytic cycle and lysogenic cycle. First, it goes with definition. A viral replication cycle that ends the host cell is what? Ends in host cell is what? Lysis. A viral cycle where the viral DNA integrates into the host DNA lies as dormant. Virus type, here they are virulent phases, these are what? Temperate phases, remember that. Next, integration. Viral DNA does not integrate with host DNA, but here viral DNA will integrate with the host DNA. That integration you will call it as what? Prophase, which is the latent phase or inactive stage. Okay. Effect on the host cell. Host cell is destroyed or lysis will take place, and here host cell will remain alive, they will divide normally. Latency. There is no latent phase over here, here latent phase will remain. And then viral DNA replication. This replicates independently using what? Host cellular machinery, but this replicates along with the host cellular machinery. And then symptoms in the host is rapid onset of the symptoms due to cell destruction. As the bacterial cell is destructed, the symptoms are very soon, but here delayed symptoms where virus may remain inactive for long time. Examples, best examples is T4 bacteriophage, virulent phases comes under lytic cycle. Here lambda phase, lambda phases will come under lysogenic cycle. They trigger the activation for, there is no need for any triggering only, but here during the stressful conditions, that lysogenic cycle will enter into the lytic cycle. Lytic cycle will be activated again during stressful condition. Burst size, when the bacteria is releasing out the virions, the burst size, it releases 50 to 200 new neurons, new virions per infected cell. So, one bacterial cell which is infected in the lytic cycle, at the end it will burst open, releasing how many virions? Approximately 50 to 200 new virions will be bursted out or released out. Here there is no virion release until the lytic cycle is induced in the lysogenic cycle. Clear, right? And then here. Now let us look into all the symptoms, diseases, nature of the infection of viral diseases in plants and bacterial diseases in plants. First, in virus. Virus nature of infection is always as an obligate parasite. Okay? Virus is always an obligate parasite, and this causes many systemic plant diseases which affect the entire plant. Symptoms of plant viral diseases include, first, like chlorosis. Chlorosis means what? Yellowing of the leaves, for example peach yellowing. Second is mosaic, that is irregular patches of discoloration, for example tobacco mosaic disease. And third is vein clearing, that is loss of color in the leaf veins, for example bendy vein clearing. And then malformation, that is stunted growth of the plant or abnormal growth of the plant. Best example is swollen coca shoot. And then breaking of the flowers, for example irregular flower coloration, for example tulip mosaic break. Okay. So, these are the symptoms of plant viral diseases. And then virion and viroids. Virion is full assembled host cell which has genetic material and protein code. Viroid is the only one which has single stranded RNA, but it does not have protein code. Okay. So, only RNA but no protein code, tiny RNA fragment. This RNA fragment will have how much? 300 to 400 nucleotides, but it does not have any protein code. Okay. Still causes the disease, but this still causes the disease. Viroids will infect what? Economically important crops like tomato, potato, cucumber and citrus. Clear, right? Any doubt anywhere? No. Common viral diseases. Just a second. Yep. Common viral diseases, mild or common diseases like what? In humans, so it is cold, hepatitis, chickenpox, influenza, herpes, warts, polio. These are all the mild or common diseases. High mortality is seen in what? Rabies, AIDS and Ebola. Okay. Long term effects of the viral diseases include polio, neonatal rubella, which leads to debility. Okay. Next, cancer causing viruses are called as oncoviruses. Okay. These are cancer causing viruses. First, like Epstein-Barr virus and human papilloma virus, this can cause cancers. Second is hepatitis B virus. Okay, this is chronic infection which may lead to liver cancer. Fine. So, first is what? Epstein-Barr virus and human papilloma virus, these are cancer causing oncoviruses. Second, hepatitis B virus, which is chronic infection, this may lead to what? Liver cancers. Next is prions. Prions, these are non-viral protein agents. Prions, you can simply define it as abnormally folded proteins. Abnormally folded proteins. Abnormally folded proteins, whereas infectious protein particles, these are the infectious protein particles, prions. Best examples for this, like they cause diseases like mad cow disease, which is popularly known as bovine spongiform encephalopathy in cows. Okay, their main target is nervous system. Okay, main target is nervous system. It has many variants. Okay, it has many variants. Same disease observed in different organisms. First is mad cow disease, bovine spongiform encephalopathy in cows. Next type of variant, that is crepey disease in sheep. Again here the target is nervous system only. Third, in humans also there is a variant which is caused by the prions. That disease is CR-Jacob disease in humans. CR-Jacob disease, which affects the nervous tissue, and the disease is caused by what? Prions. Okay. These are the different viral diseases. Viroid diseases. Viroid diseases, best example is potato tuber spindle disease. Potato spindle tuber disease. And next, prions is best example, mad cow disease, crepe disease in sheep, and CR-Jacob disease in humans. These are all about the common viral diseases. Now let us look into bacteria. Okay, bacterial structure and its types. Physiology we will look into tomorrow. So bacteria abundance. Bacteria are abundant among most of the organisms on the earth. Like bacterial distribution, we can say it is ubiquitous in distribution or cosmopolitan in the distribution, wherein these are found in soil, water, air, food, and inside the living organisms. Historical contributions include, like Anton van Leeuwenhoek in the year 1674, he first to observe the bacteria. He is the first person to observe the bacteria. Second is Ehrenberg in the year 1829. He has coined the term what? Bacteria. All this historical background or the historical contribution is important for us. Okay. Louis Pasteur in the year 1850, he showed that bacteria acts as what? Chemical factories which cause changes in the nature. Okay. Louis Pasteur has shown the bacteria will act as what? Chemical factories which cause changes in nature. He is known as father of what? Bacteriology. Louis Pasteur is known as what? Father of bacteriology. Robert Koch in the year 1870s linked bacteria to infectious diseases. He has linked bacteria to infectious diseases via germ theory of disease. Okay. Yeah. Bacteriology. Due to continuous research, the study of bacteria became a branch of biology, that is bacteriology. What is the ecological distribution? As I told you, they are ubiquitous in distribution. They are present everywhere. In the extreme environments like Arctic snow, volcanic ash, hot water, sulfur springs, and they can withstand extreme cold, heat, drought conditions. That is because of their cell walls. So if you see bacteria, we have majorly different types of bacteria like archaebacteria, eubacteria, methanogens. All of them are present in wide diverse atmospheres. That is because of their cellular adaptations. Okay. So here the outnumber of all other organisms in terms of sheer population. Fine. Role of bacteria. Some are parasites of plants, animals, and humans. Some from mutualistic symbiotic relationships. Okay. Some are from mutualistic symbiotic relationships. So this is about bacteria. And then morphology of bacteria. Bacteria, they are very small. They can only be seen under the light microscope. Okay. The size ranges from what? 2.5 to 5.0 micrometers in length, and breadth is about, that is width. Okay. Width is about 0.5 to 1 micrometer in width. What is the shape? Usually it has a rigid cell wall. Shapes can be cocci, which is spherical, bacilli rod shaped or elongated, spirillum helical, vibrio is comma shaped. Of these 4 different shapes of bacteria, which is most pathogenic? The bacilli shaped bacteria are most pathogenic. Okay. And pleomorphic. What is meant by pleomorphic bacteria? Few bacteria are pleomorphic. They change their shape based on the environment and nutrients. Okay. Those bacteria which vary in their shape or they change their shape according to the environment and nutrients, that is considered to be pleomorphic. Okay. So the best example is Acetobacter. This is about the shapes of bacteria. And then some spiral forms, for example spirillum, are flexible and are called as what? Spirochetes. Okay. Spiral forms are what? Spirochetes. Others occur as filamentous chains, for example Bigiatova. Okay. Some in the spiral form spirochetes, some occur in the filamentous chains Bigiatova. Bacterial arrangement, coccus forms, like we have coccus, bacilli, vibrio, we have these many types, right. In cocci also, based on their association, spherical association, we have different types of coccus. Monococcus single type, diplococcus, pair of cells, tetracocci, group of 4 cells, streptococci, a linear chain of 8 cells like this, linear chain of 8 cells, staphylococci. This association will form a cluster or a bunch like this, okay, and sarcinae, cubes of 8 cells. Sarcinae is what? Cubes of 8 cells. Fine. These are different types of arrangements in only coccus forms. Can you see? Coccus, diplococcus, staphylococcus, streptococci linear chain, sarcina cube of 8 and a tetrad. And then bacterial arrangement of bacillus type, which are mostly the pathogenic bacteria. So, here this bacterial type include like monobacillus, diplobacillus and streptobacillus. Monobacillus is single bacillum, okay, diplobacillus 2 rods, okay, and streptobacillus is a chain of bacilli, rod shaped cells. So, this is about different shapes of the bacteria. In our next class, we will be looking into different types of bacteria based on their cell wall and bacterial structure, physiology, nutrient diversity and reproduction. Reproduction where we will be dealing with conjugation, transformation and transduction. Clear, right? So, that is all for today. We will meet in tomorrow's session with the above concepts which I have told. Okay, thank you.